Hi, everybody. We are back and we are picking it up again on chapter 14. And just to recap a little bit, we already talked about how how we work monohybrid and dihybrid crosses. So we talked about simple Mendelian genetics, how he studied the garden pea, and how he used that to formulate simple mathematical probabilities of what's a chance of different offspring showing up based on what the traits the parents have. So again, we were looking at monohybrid crosses where we're just looking at single trait crosses where we'd have that 2 by 2 box, and then dihybrid crosses where we're looking at two different traits, meaning the parent boxes are each going to have two different genes and the offspring would have a combination of four alleles. So now we are picking it up on page the 83 of the course outline and looking at going beyond Mendelian genetics. So there are many different alternatives and combinations and lots of things we can't even really explain when it comes to inheritance. But so now we're going to go beyond Mendelian genetics and look at some different cases. So here's three listed here. First of all, when alleles are not completely dominant or recessive. So instead of one being dominant over the other, when they both show up, they end up blending. When a gene has more than two alleles, instead of just one dominant, one recessive, you might have two dominants and one recessive, or something along that line. And then third, when a gene produces multiple phenotypes. So one gene can influence many phenotypes. Remember, phenotypes refers to the physical appearance. So we have names for these different types of genetic situations. Complete dominance is what we've looked at so far. This occurs when phenotypes of the heterozygote and dominant homozygote are identical. So in this case, we just have one dominant, and that shows up over the recessive condition. In codominance, this is a different situation where they're both dominant, so you might have two dominant alleles that affect a phenotype. And then the third case is incomplete dominance, where neither one is dominant over the other, and instead, in the heterozygous condition, they have a blending effect. So we're going to begin, first of all, looking at what we call multiple alleles, where we have more than just two alleles affecting a trait. And in this case, there are three alleles that influence blood type. This is also a situation of codominance. Codominant meaning that there are two dominant, one recessive. So when it comes to the ABO blood groups, A and B are both dominant over O, which is a recessive condition. So what are these blood types and how did we figure out how you inherit them? And just a little history of blood groups and blood transfusions. So in the early days, in the 1800s and even before, when people were bleeding to death, somebody came up with an idea, hey, how about if I give you some of my blood, see if that'll work? And sometimes it worked and sometimes it didn't. And when it didn't work, those people actually ended up dying because their blood clumped or agglutinated. And when they did autopsies, they would find that these people who died from that blood transfusion had blood that looked almost like jello. So there were lots of experiments done before they finally figured out what was going on. And as it says here, the experiments with blood transfusions had been carried out for hundreds of years. Many patients died, and it wasn't until 1901 when the Austrian Karl Landsteiner discovered human blood groups that blood transfusions became safer. And he found that mixing blood from two individuals can lead to that blood clumping. And the clumped red blood cells can crack and cause toxic reactions, and that can be fatal. So that's where it all began. And this was based on an immunological reaction which occurs when the recipient of a blood transfusion has antibodies against a donor's blood. And so what he did was, in order to determine these blood types, he then actually started mixing bloods of different types. And he found that on the red blood cell, there are certain proteins. And the first protein they discovered, they called protein A, and that was blood type A. If you had that protein, they would call you type A. And then they found B. And then they found some people had both, so they called them AB, and some people didn't have any, and they called them O or 0. And so, looking at these blood groups, the differences in human blood are due to the presence or absence of certain protein molecules, and those are called antigens. And then there are also antibodies. The antigens are located on the surface of the red blood cell, and the antibodies are in the blood plasma. And so what happens then is if you are, say, type O blood, you don't have any of these antigens, and somebody gives you type A, your body perceives that A protein as foreign and attacks it. And so you have an immune reaction, and that causes the clumping, and the clumping, of course, then leads to all the problems I mentioned already about the blood agglutination. So individuals have different types and combinations of these molecules, and the blood group you belong to depends on what you inherited from your parents. So that's what we're going to look at next. Now there's a couple different ways you can set these up. First of all, remember the ABO blood groups, that's just the first one they discovered. Later, they discovered another protein, and they discovered it using a rhesus monkey, and they called that RH. So RH factor is a separate inheritance. And then, since then, they have found hundreds of proteins on the red blood cell. And so today, rather than if you're in an emergency situation, they might give you type O because it doesn't have any antigens, and they might give you RH negative because negative means it doesn't have antigens. But if you know you need blood, instead they will do what is known as a cross match. They'll take your blood and mix it with the donor's blood before they give it to you to make sure there aren't any kind of incompatibilities, because there's so many different kinds out there. But the ABO blood groups and the RH still cause the most problems, and we'll talk about why here in a minute. Remember, the ABO blood group is a case of what we call multiple alleles, both, and also codominance. A and B are both dominant over O. So when you look at the different genotypes and phenotypes, here they use the letter I and attach the A's and B's, and or just leave it lowercase to represent O. You can do it this way, or you can just drop the I. The I is for immunoglobulin, which is the protein that I mentioned. Or, if you want, you could just put A's. So if you are, here's your phenotypes. You can either be type A, type B, type AB, or type O. If you are type AB, since A is dominant over O, I don't know if you are AA or AO. As long as you have one A, you're going to be phenotypically called type A, but you could be homozygous or heterozygous. So this is showing the red blood cell with the protein A attached. If you are type B, again, B is dominant over O, so I don't know if you're homozygous or heterozygous. If you are homozygous, you inherited a B from each parent. If you're heterozygous, you inherited a B from one parent and nothing, or O. O actually means 0, nothing, from the other parent. So you would be heterozygous for type B. If you are type AB, I know your genotype has to be AB because you had to get an A from one parent and a B from another for both of those to show up. So remember, AB, this is co-dominant. Both A and B are dominant, and they both show up. And only if you have type O would you have none of the above. So O literally means 0, as in nothing. So this is the homozygous recessive condition. So I usually show it as OO. They're showing it as lowercase I. So you could do whichever one you want. So when it comes to blood typing and doing Punnett squares, then, when you look at the phenotypes listed here, you can be type A, type B, type AB, or type O. Remember, if you're type A, we don't know if you're homozygous or heterozygous. If you're type B, we don't know if you're homozygous or heterozygous. But if you're type AB, we know your genotype has to be an A from one and a B from another parent. And if you're type O, you have to be homozygous recessive. That is, you inherited O's from both parents. Because again, if you had an O and an A from one parent, you would be type A. Remember, A and B are both dominant over O. Now, one of the things that we do in lab is we actually type blood, but we can't use real blood anymore. So we use simulated blood. In the lab, we actually have some simulated blood from um, John Doe Sr., John Doe Jr., Jane Doe, and Wiley Smith. And you have to decide who's who. You have to type their blood and determine who's the daddy. So here's the thing about blood typing. Blood typing can disprove paternity. That is, it can show who's not the daddy, but it can't prove paternity. And so to do that, you would have to actually have a DNA test. But here's how it works. So what you do is you'll take a slide and you'll put an antigen for A and an antigen for B on that slide, and then you mix the blood. And if it's type A, it's going to have an antibody antigen reaction and clump. So if you have clumping like you see here on A, it's type A. If you have clumping as you see here on B, it's type B. If you have clumping on both, it's going to be AB. And it contains agglutinogens for A and B. And if now you can see type B only has clumping on B, type A only has clumping on A, and notice type O has neither, 0. Okay, so once again, clumping on A is A, clumping on B is B, clumping on both is AB, and no clumping is O. So let me back up and do a couple Punnett squares. So if you have noticed again in your course outline, you have some practice problems. By now you should be able to do the problems on page 87 and 88 dihybrid crosses. Hopefully you practice those. But if you look over on page 88, we see some codominance multiple allele problems, which those you should practice here for blood typing. But I'll show you how to do one. So a good question when it comes to blood typing is, can a type O person and a type AB person have a child with either type O or type AB? In other words, one parent is O, one parent is AB. Can they have kids that are the same as them, type O or type AB? And usually when I ask this in a classroom, students generally think, yeah, why not? Well, here's why not. So let's talk about this. So let's say we have a parent with, we're going to cross a type O with a type AB. Now remember, with these, we know the genotypes. Type O can only have OO, and type A can have an A and a B. So we know these genotypes. So we set it up just like a regular Punnett square. We put one. This is a monohybrid cross, so make sure you just put one allele in each box, an A in one and a B in the other. In this case, you can mix letters. And then type O, remember, only has O's. Bring these down and across just like you would a typical monohybrid cross. And notice they could have type A, heterozygous for A, and type B, but they cannot have type O or type AB. Okay, so let's try another one. Let's say if you know you have one parent is type A and one parent is type B. So one parent is type A. Sometimes when you do these problems, you just fill in what you know. Type A, I don't know if they're homozygous or heterozygous. And type B, I don't know if they're homozygous or heterozygous. But if they have a type O showing up, what do they have to be? What would they have to have here to have a type O show up? They would have to also have an O. And if we bring this down and across, you can see that all 4 blood groups can show up in that family. So that situation, you really can't prove or disprove, that is, who's the daddy. So when you do this lab, and I'll be talking about this a little bit later, when you're trying to disprove paternity, just show what kinds of combinations those parents can have. And when in doubt, if you don't know if they're homozygous or heterozygous, just go ahead and put in a heterozygote because that will give you the most combinations and the most possibilities if you're trying to see what's the possible outcome for those children. Okay, so that's the ABO blood types. When I teach this, usually I get questions also about Rh factor. And here's the thing about Rh factor. As I mentioned, this was another blood type. It's a different gene. It's inherited completely independently. So whether you're A, B, or O has no influence on whether you're Rh positive or negative. But this is a typical one gene, two different forms, one dominant, one recessive. It's not multiple alleles, it's not codominant like we saw with the ABO. So if you told me that your mom's O positive and your dad's, I don't know, B negative, what's the chance of having kids with different combinations? I would have to do two separate Punnett squares because they're inherited totally different and separately. So when you do an Rh factor, here's your possible phenotypes. You can either, if you have that protein, we say you're positive, and if you don't have it, we say you're negative. There's your phenotypes. If you're positive, though, which is dominant over negative, I don't know if you're homozygous, that is, you inherited a positive from each parent, or if you're heterozygous, that is, you inherited a positive from one and a negative from the other. Either way, phenotypically, we say that you are positive. If you are negative, I know your genotype has to be only negative from each parent because, again, that's recessive. So just like all recessive traits, it has to be homozygous to be recessive. That's all you can have. So here's the problem with Rh incompatibility situations. This is a case where a father is positive and a mother is negative for Rh. And normally, these blood antibodies don't cross over the placenta. But the problem with incompatibility situations is when the Rh negative mother is carrying her first Rh positive fetus, the Rh antigens from the developing fetus can enter the mother's blood during delivery. It's only during delivery when that placenta breaks away from the mother, a little bit of baby's blood enters mom's blood. That's the only time the blood mixes. And when that happens, that sensitizes her to these positives, and so she builds up these antibodies. And so as you see here, these little green Ys are representing antibodies which stick around, and if she gets pregnant again with another positive baby, the chance that these particular antibodies are small enough to cross the placenta. And so during the entire pregnancy, they move across, they attack the baby's blood, and the babies used to be born either anemic or sometimes they even died because of this. So it was they needed blood transfusions, all kinds of problems. But the good news is if you know you are a female who's negative and your husband or father of the child is positive, and even if you don't know if the baby's positive or negative, and I'll show you a problem to figure out what's the chance in a minute, there is a drug that they can give you, and they just give it normally to pregnant women who have negative type blood. And I always forget if it's Rogam or Rogaine. One of it's Rogam, right? Rogaine is the one for hair loss. So I always figure if they mix up these drugs, you'll have a hairy, anemic baby. But Rogam, I'm hoping, yeah. Anyway, they give it to you right in the beginning of the first trimester because there's a higher chance of miscarriage at that time. And then they give it right before delivery so that it tricks your body into thinking you're positive so that you don't build up these antibodies to attack the baby. So it's an easy fix if you know that the female is negative. So it's very important, ladies, to make sure you know your blood types. You can donate blood. They will tell you your blood type for free. And that way you can be sure if you are negative that you can keep an eye on this. But let's go back to the drawing board and do a problem with Rh incompatibilities and show you what's the chance of inheriting this if you are a negative person. So as I said, this is going to be done completely different and separate from the ABO blood types. And I'll just go ahead and put up a chart here. You can see that if we're looking at the genotypes and the phenotypes for Rh, so phenotypically you could either be Rh positive, or let me just write that as positive, or you could be Rh negative. Those are your phenotypes. But genotypically, it's just like a typical monohybrid cross in that the dominant one will show up over the recessive. In this case, Rh negative is recessive. So for Rh positive, you could either be homozygous, you inherited a positive from each parent, or you could be heterozygous and you would still be said to be Rh positive, just like any dominant trait. For Rh negative, it would be just you only have negatives here. Okay, so looking at a problem, let's do a Punnett square. We set this up just like, again, a typical monohybrid cross. And let's look at, say, a negative mom, so we know what her genotype is. And let's say dad's positive, and even at best, if he's heterozygous, what's the chance of her having a positive baby? And so we bring these down and across. And at very best, it's 50-50. Now, if she has a negative baby and she's negative, no problem, right? But if she has a positive baby and she has a 50% chance, then she has to worry about Rh incompatibility disorder. And so she would need that drug. So that's really all there is to Rh Punnett squares, is you just set them up. And remember, you have to do them separately because they're inherited separately. Okay, moving on to incomplete dominance. Now, incomplete dominance is a case where neither allele is dominant over the other. And in the heterozygous condition, they actually blend. So this is the case, for instance, in snapdragons, these flowers you see here, where the red flower color and the white flower color are incompletely dominant. And in the heterozygous condition, they show up as pink. And so when you do these problems, now the way your author does them here, he uses the letter C to represent the gene for flower color, and then he attaches an R for the red ones and a W for the white ones. And you can do it that way if you want, or you can always just, what I do is I always, anytime I see, when I read a question and I see it's incompletely dominant right away, I write down A1s and A2s. And that way I don't get mixed up by accidentally putting a big A and little a. Because if I put a big A and little a down for my key, that tells me I'm dealing with dominance. And this is not a dominant situation. This is, again, incomplete dominance. So when you see that word, write down A1 and A2. And it doesn't matter which one's which, because they both blend in the heterozygous condition. So I'm just going to say A1 is red and A2 is white. Now I'm going to cross a red and a white flower, but here's my possible genotypes. I can have homozygous for A1, in which the flower would be red. I could have homozygous for A2, in which the flower would be white. Or I can have one of each. And then what color is it going to be? Pink, right? Okay, pink. So let's cross it. What's the chance of crossing a red and a white and getting a pink? So if I cross a red and a white, again, I do a 2x2 box. Again, all of these are very similar to monohybrid crosses. And if it is a red, I know that's going to be A1s. We put one allele in each box. In dihybrids, remember, you put 2 alleles in each. So one allele in each box for red, and then the white would be A2s. Bring these down and across just like your typical monohybrid cross. And notice that they are all pink. Okay, these are all pink. Do this because I need more room. So this would be 100% pink. This is the F1. But what if I wanted to carry the F1 out to the F2? So these are the F1s. I could cross any of those pinks. They're all the same, so it doesn't matter. Bring these down and across, just like your typical monohybrid cross. And here is your outcome here. So now you have a 1:2:1 ratio. In other words, here you have 25% A1s, which are red, 50% pinks, and 25% I did that wrong. That should be A2s. Oopsie. Somebody should have told me I did that wrong. So weird not having students. Okay, 25% A2s, which are white. I'm sure some of you were going, you did that wrong. Anyway, just testing, see if you're following me. So again, we could call that a 1:2:1 ratio if we wanted. Okay, so the important thing to remember on incomplete dominance is don't put big A and little a, because that's going to make you think one's dominant over the other. So always put A1 and A2. Other than that, just do it exactly like your typical monohybrid cross. All right, on to polygenic inheritance. Polygenic. Poly means many, and genic means gene. And in this situation, this is a case where many genes influence one trait. For example, skin color. We talked about skin color. It's not just black and white. We don't just have a dominant allele and you're black and a recessive allele and you're white. That's it. No. In skin color, there's actually many genes that influence it. And the more dominant alleles, the darker you are, and the more recessive alleles, the lighter you are. And so when you look at something like this, if we see that there are, say, 3 genes that influence skin color, and we see that would be 6 alleles, right? For every gene, there's 2 different forms. If you inherit all recessive alleles, we might say this person is white. That's one extreme. Or all dominant alleles, we might say this person is black. But what if you get half and half? You're somewhere in the middle, medium. And then this person's got more recessive than dominant, so maybe that person's light. This one's more dominant than recessive. Maybe that person's dark. So again, we don't have just black and white here. And here is a clip on how this is inherited.
To fly a rocket ship, you need to be an optimist. No astronaut.
Not all of our traits are controlled by the inheritance of a single gene. Our height, weight, and the color of our eyes and skin are all controlled by more than one gene, and are thus referred to as polygenic traits. That eye and skin color are controlled by more than one gene, respectively, is reflected in the fact that there is such a variety of gradations of eye and skin color. People don't just have blue eyes and brown eyes, but rather there are blue eyes, green eyes, hazel eyes, light brown eyes, black eyes, etc. The same is true for skin color, and perhaps has even more variations. Both eye color and skin color are the result of the presence of a pigment called melanin. If melanin production were controlled by one gene, and an individual had two recessive alleles and no melanin was produced, they would have light skin. If they had one dominant gene and one recessive, their skin color would be of medium tone. And if they had two dominant alleles, they would be extremely dark. However, we see a much greater variation in skin color than this among individuals. This is the result, biologists believe, of at least 3 incompletely dominant genes controlling skin color. These combinations among 3 different genes allow for 7 different shades of skin among individuals. Likewise, biologists believe 2 or 3 genes control eye color, explaining all the various shades of eye color, as opposed to just one gene controlling eye color, which would result in only 3 different eye colors, say blue, light brown, and brown. But as we know, there is a much wider variety of eye color than this. There are also a large percentage of infants who have blue eyes that later become a darker shade, such as hazel or brown. And this is probably due to the fact the infants have not started producing melanin in their irises yet.
Okay, so polygenic inheritance can also be seen in body height. For example, if I asked you right now if you are tall to go to the right side of the room and you are short, go to the left side of the room, I'm sure there'd be quite a few of you that don't know where to go. And that's because we have something called continuous variation when it comes to height. Just like we saw with skin color, it's a bell curve. There are not very many very, very short people and not very many very, very tall people, but most of us are somewhere in between. And this is just showing, for example, if body height were all within the 5 to 6 foot range, which of course it isn't. But again, when we're looking at how many people are just 5 feet tall, not too many. How many are 6 feet? Not as many as well. So we see this continuous variation. So think about polygenic inheritance as a cumulative effect. The more dominant alleles, the more melanin, the darker your skin, the more recessive alleles, the less melanin, the lighter your skin. Well, when it comes to polygenic inheritance and body height, you can think of it as sort of adding inches of height. And as an example, let's just say there are 4 genes, again, polygenic, many genes, gene A, gene B, gene C, and gene D, that all code for body height. And let's say if you inherit gene X, you have a base height of 5 feet tall. So if you inherit the dominant allele, you get an added ½ inch. A recessive allele, no additional height. So if we ask ourselves, and I should have put an X right here, so this person is at least 5 feet tall, they inherited X. How tall is someone with this genotype? Remember, every dominant allele adds an extra ½ inch. Every recessive allele adds nothing. So for every dominant allele, add an extra ½ inch, and this person will be 5 feet what? How many inches? Give you a minute to think about that. How many dominant alleles? Add them up. ½ inch per dominant allele. All right, let's see if you got it. How many got 5 feet 3 inches? So, of course, the 5-foot base height, but the 3 inches comes from there's 2 A's, that's a half and a half is 1. A B here and a C here, that's 2 more inches, and a C and a D here. So we have 6 dominant alleles, that would be 3 inches. How tall would the tallest person be if they had all dominant alleles? The very tallest person would be what? Exactly, 5'4". Because if every one of these were dominant, that'd be 1 inch, 2 inch, 3 inches, and 4 inches. How tall would the shortest person be if they had all recessive alleles? They would just be 5 feet, right? So in this case, you don't do a Punnett square. You just read genotypes and decide, adding each one. So that's how I have for polygenic inheritance and course outline, just giving you some genotypes and telling me who's the darkest, who's the lightest, who's the tallest, who's the shortest, and it's just based on how many dominant alleles there are. Okay. Now, hair color. This is a confusing one. How do we get all these different hair colors? Let me show you this clip and we'll explain.
Good to see you.
Yeah, hey, what's with the polka dots?
No, what do you mean?
Polka dots are out. It's polka squares now.
Oh, yeah.
Sometimes you're right in the middle of cleaning out the drain in the shower and you start pondering questions like, why is my hair a different color from my mom's hair, or my neighbor's hair, or my roommate's disgusting, soggy, 3-foot-long wolf-tail drain wad? What's the real difference between blonde hair, black hair, red hair, and everything in between? The main structural ingredient in human hair is a protein called keratin. It's what your hair and fingernails are made of, but also what's behind the silky sheen of wool, bear claws, and horse hooves. Don't you just want to run your fingers through those hooves? Just me? But keratin on its own is not very colorful, and if all humans had on our head was keratin, we'd look like 18th century French aristocrats in powdered wigs because we'd all have the same sort of white, colorless hair. But keratin is not the only ingredient in human hair. To create natural color, you need to add pigment, and this is done by cells in the skin called melanocytes. These melanocytes create the natural pigment known as melanin and deliver it to the cells that create keratin for your hair. Now, this melanin comes in two varieties, eumelanin and pheomelanin. Eumelanin is a dark pigment that gives hair a brown or black color. Pheomelanin is a lighter pigment that gives hair a red, orange, or yellowish color. Both of these are present in varying degrees, so a person might have a little of each, or a lot of one, or almost none of the other. So someone with black or dark brown hair probably has a lot of eumelanin, not to brag. A redhead has a lot of pheomelanin, and blondes don't have very much of either one. So what happens when we get older and start to go gray? Well, you can probably guess. Over time, melanocytes start to die off, and any new hair that grows has less pigment, so it looks gray or white. But you might be asking, what determines the eumelanin to pheomelanin mixture to begin with? Who writes that hair color recipe? Primarily, it's your genes. For example, the melanocortin-1 receptor, or MC1R gene. When the protein associated with this gene is active in the melanocytes, it stimulates them to make eumelanin, the pigment that colors black or brown hair. When MC1R is not active in the melanocyte cells, they make mostly pheomelanin instead, and hello Weasleys! But the MC1R gene is not the only genetic factor that controls hair color. Like most of your traits, hair color is actually affected by more than one genetic variable. So, that's the scoop on hair color. But hey, what's the weirdest thing you've ever noticed about human hair? Let us know in the comments, and if you ever want to learn more about the strange science of the human body with brain cells.
Okay, well, so you can see how complicated it is when it comes to hair color. Again, this is another example of polygenic inheritance. So we see, just like with eye color, it's not just a matter of 2 or 3 genes influencing that trait, but how they're combined and how they're expressed. And there's environmental factors, both external and internal environmental factors I'll get to in just a minute. So it gets very, very complicated. So if somebody wants to know, well, both parents have blue eyes, am I going to have a blue-eyed baby? Not necessarily. It just really depends. So eye color is even more complicated. Here's a clip on that one.
Although for one particular gene, say for eye color, the relationship between alleles may be clear, so that, for example, brown eyes may be dominant to blue eyes. The phenotypes observed in the population, however, may be the result of the interaction between this first gene and a number of other genes. So, for example, there may be a second gene which then determines how much brown pigment is made. So the two individuals could both have brown eyes, but nevertheless result with different hues. A third gene could perhaps determine the exact hue of the brown. So the brown hue made by one person's body might not be identical to the brown in a second person's body. Another gene might be responsible for whether brown pigment is made throughout the iris or only in spots. And yet another gene might cause pigment to be concentrated around the pupil. Then finally, there may be other genes which are capable of masking this first gene, so that even though an individual possesses alleles of a gene to give them brown eyes, the alleles of a different gene can mask those so that they will then possess blue eyes, but be able to pass down the gene for brown eyes to their children. And so, because multiple genes can interact to produce a phenotype, 6 individuals who all possess the same alleles for brown eyes could nevertheless possess different colors in their iris.
So usually about this point, somebody asks me about people with two different color eyes and what is that all about? And that has to do with something known as genetic mosaicism. And mosaicism, that word mosaic means an intricate pattern. And it involves the presence of two or more populations of cells with different genotypes in one individual who has developed from a single fertilized egg. Now, if it's in the somatic cells, remember the somatic cells are our body cells. So they don't have anything to do with reproduction and passing on this trait. But it could be that those particular cells may have some kind of change. And as it mentions here, it could be that there was problems with mitosis, because remember, somatic cells are the ones that are involved in mitosis, a mutation, and so wherever that mutation is, only those cells that have the mutation then would show up that color. So it could be in the iris itself, the colored part of the eye, that you get just certain cells that have the mutation showing up as a different color. Or it could be, as you see here in heterochromia, which the iris is multicolored or a completely different color, as you see here. So sometimes it's genetic, sometimes it's a growth or an injury. It could be a lot of different things that cause that. So not necessarily something, even if it's genetic, if it happens in the somatic cells, it's a change in DNA in those cells aren't going to necessarily be passed on. So remember, the only thing that is passed on is what happens in the egg cells and the sperm cells. So that's the situation with that. And those are some examples of polygenic inheritance. Now, what if we mix it all together and we look at things like, what if we have a situation where things are codominant and polygenic and have a blending effect? Then we get really, really complicated. And that's what we see oftentimes in what is known as epistatic interactions. And that can be seen in coat color of Labrador retrievers, as you see here. In labs, you can have your classic black lab, you can have a chocolate lab, you can have a yellow lab, and you can have a white lab. And so what happens here is coat color is, as you see here, there's a try this, by the way, in your course outline. And this is a good example of a type of problem that you would see on epistasis. And so in this one, you can, I'm looking for the page number, there it is, on page 84. Um, there are, in this example, there are 3 genes that affect coat color in Labrador retrievers. So gene D influences whether melanin is produced at all. And if you don't have any melanin production, then the dog is going to be albino. And that stops there, because if you don't have any melanin production, it doesn't matter what you inherit as far as what melanin is deposited or how much melanin, because you don't have it in the first place. So this gene D cancels out everything else in the homozygous recessive condition. But as long as you have a big D, whether it's homozygous dominant or heterozygous, then you can go on to the next gene. And the next gene says, well, how much is deposited? As long as you have a big E, then the melanin is deposited. But in the homozygous recessive condition, everything after that is blocked. Deposition is blocked, you end up with a yellow lab. And then the third thing is looking at the quantity of melanin. Assuming that you're producing melanin, you're depositing melanin, then you can say, well, how much? And here, as long as you have a big B, the dog is black. In the homozygous recessive condition, the dog would be chocolate. So when you look at these genotypes, again, there's no Punnett square here. You just look at the genotypes and determine what color the dog is. If you look at the letters here, you have to start with gene D because that tells you whether you're even producing it. And if you're not, if you're in this homozygous recessive condition, everything else is blocked. I could cross out since this is homozygous recessive, that dog right there is going to be albino. It's not even producing melanin, so we cross out the E's and the B's, and that's that. Now, for number 2, is the dog producing melanin? Yes, he's got a big D, but is he depositing it? No, because he's got the 2 little E's here. That when deposition is blocked, that dog then is going to be yellow. It doesn't matter what he has for B because he's not even depositing it. The third one, is he producing it? Yes, he's got a big D. Is he depositing it? Yes, he's got a big E. So now the third question is how much? And since he's got a big B, that dog's going to be black. And then the last one here, if he's producing it, yes. He's depositing, yes, but he's got little b's, that means he is chocolate. So again, you have to read these backwards. So this is an example of epistasis, which means that genes can actually block the effect of others. So even if you inherit a homozygous dominant situation for quantity, it doesn't matter because you're not even producing it. So these are, again, so many variables that influence things like eye color, skin color, coat color, and animals. All kinds of traits are like this. A lot harder to explain inheritance with this. So to summarize what we've looked at so far, we've looked at complete dominance. That's where we started, where things like purple was completely dominant over white. So that's a case where you have one gene, 2 different forms. Then we looked at incomplete dominance, where there's a blending effect. Remember, when you see this, you put down a A1 and an A2. You don't want to put big A and little a down, or you'll think you're talking about complete dominance. Codominance, remember when you can have more than one dominant. In the case of the ABO blood types, both A and B are dominant over O. So if you inherit an A from one parent and a B from another, both show up. This is also a case of what are called multiple alleles. That is, there's more than 2 alleles involved in this inheritance. In this case, there's 3. 1 for A, 1 for B, and 1 for O. And then pleiotropy, this is where one gene is able to affect multiple phenotypic characters. We're going to talk about that next. A good example is sickle cell disease, so we'll come back to that one. Epistasis, we talked about this with the Labrador retrievers, and coat color. Keeping in mind that one gene can block the effect of others. And polygenic inheritance, this is where one gene is influenced by, or one trait is influenced by many genes. Poly means many, genic means genes. So you can have many genes. In the case of skin color, maybe 3 genes influencing those traits. And that wraps up some of the exceptions to Mendelian genetics. One other note I want to mention here is that the environmental effects can influence how genes are expressed. This is a great example of an Arctic fox in the winter compared to the fox coat color in the summer. Why does it have a white coat in the winter? He's lucky he does because he's much more camouflaged with white in the winter and multicolored in the summer. And that's because the genes that he's inherited are there, but they're influenced with enzymes. Remember, you can't produce melanin or anything else without the enzymes needed for those metabolic pathways. And those particular enzymes in the Arctic fox are affected by temperature, as most enzymes are. And so when it's cooler, the enzymes aren't working as much and they're not producing melanin and the coat turns white. In the summer, when it gets warmer, it activates those enzymes that are heat sensitive, and you can see the melanin production comes back, and the Arctic fox is well camouflaged for that. So other examples, when you look at eye color, have you ever noticed some people's eyes seem to be chameleon-like? They will change based on what they're wearing. What do you know about light? Remember that it is absorbed and reflected at different wavelengths, and so it really is a matter of light and how it's absorbed and reflected in your iris, what color your eyes look like at different times. So there's that. So there's internal effects, as I mentioned with these enzymes. There's external effects as well, like sunlight. And our own hair changes color as we age, just like our skin lightens, our hair lightens, our eyes lighten, as those genes that influence those traits start getting turned off and we stop producing melanin. So many, many environmental effects on alleles. All right, now we're going to switch gears a little bit and look at the behavior of recessive alleles in terms of genetic disorders. So now we're going to change gears and look at genetic disorders. And we are over on page 85 of the course outline. So here's the thing about inbreeding. I mentioned this at the beginning. And when you have a lot of inbreeding, then that increases the chance that two recessive alleles will show up. And when that happens, that's when genetic disorders show up. So far we've talked about when things go right. What about when things go wrong? And how do we explain some of the genetic diseases that we have? So we want to start with looking at the behavior of recessive alleles. These recessive disorders are inherited and show up only when individuals are homozygous for the allele. So just like any recessive disorder or recessive trait, that is, they're only going to show up when you inherit one or you get two copies, one from each parent. So some parents or some people only inherit one faulty allele from their parent, and in that case we call them carriers. So they are heterozygous individuals who carry a recessive allele, but they are phenotypically normal. So most individuals with recessive disorders are born to carrier parents. That is, the parents are carriers, they have a faulty allele, but they don't know it because they appear to be normal. And all of a sudden they end up with a kid who's, say, albino. So albinism is an example of a recessive condition. And that will show up again. Usually normal pigmented parents all of a sudden have an albino child, didn't know they were carriers. Here's how this is inherited.
Cystic fibrosis results from the failure of proteins in cell membranes, which allow chloride ions to pass out of the cell. The inability of the chloride ions to cross the cell membrane properly also affects the transport of sodium ions and of water in and out of the cell. The result is that cystic fibrosis victims, rather than having the thin, free-flowing mucus of most individuals, have mucus that becomes thick and sticky as a result of the lack of water in the mucus. About 1 in 20 Caucasians carries one of the recessive alleles for cystic fibrosis, and this results in 1 in 2,000 Caucasian babies being born with cystic fibrosis, which translates into 4 or 5 babies born with the fatal disorder each day in the United States. In recent years, the knowledge of cystic fibrosis and advances in medicine have improved the quality of life for people with the disorder and substantially lengthened their life expectancy. This is the result of physical therapy, breathing exercises, modern antibiotics, and special diets. But eventually, the disease does prove to be fatal. Sickle cell anemia results from just a single amino acid being different in the polypeptide chain that forms hemoglobin molecules. This slight change causes the abnormal hemoglobin molecules to be less soluble and form crystals within red blood cells. The crystals distort the red blood cells into a sickle shape that gives the disease its name. In addition, the formation of crystals prevents the hemoglobin from carrying oxygen effectively, and make sickle cells rigid so that they often block capillaries. Blocked capillaries can lead to pain, fever, and weakness in affected areas. Clots formed by sickle cells can break off and cause stroke in even very young children. Like cystic fibrosis, the life expectancy of sickle cell patients is being substantially lengthened as a result of a variety of therapies, including transfusions, antibiotics to treat infections, and drugs that increase the oxygen-carrying capacity of the red blood cells. Sickle cell anemia currently affects about 1 out of 500 Americans of African descent, while about 8% are heterozygous carriers of the disease. In parts of equatorial Africa, 1 out of 25 individuals have the disease, while about 30% are carriers. Given the severity of sickle cell anemia, one asks why natural selection hasn't eliminated the sickle cell alleles. The answer may lie in the fact that being heterozygous for the sickle cell trait affords protection against an often fatal form of malaria. This form of malaria, like all forms of the disease, is caused by protozoans carried by mosquitoes that invade an infected individual's blood cells. Abnormal hemoglobin, which accounts for about half of the hemoglobin in heterozygous individuals, appears to hinder the development of the disease-causing protozoans in an individual's blood cells, and thus often turns out to be the difference between life and death in heterozygous individuals infected with this form of malaria. In most other regards, such as health and physical prowess, the 50-50 combination of normal and abnormal hemoglobin appears to leave heterozygous individuals largely unaffected. In fact, many world-class athletes are heterozygous for the alleles that cause sickle cell anemia.
So again, we're looking at right now recessive disorders, but here's the thing. When people usually have genetic disorders in their family and they want to know what's the chance of them inheriting it, I really need to know two things. Is it a dominant or recessive disorder? And is it on the autosomes or on the sex chromosomes? So we have 46 chromosomes, right? And we have 23 pairs. And 22 of those pairs are what we call autosomes. And pair number 23 are the sex chromosomes, and they're inherited. Any genes on those are inherited differently in men versus women. So we'll come back to those. But for now, we're going to be talking about autosomal recessive disorders, so it doesn't matter what your gender is, whether you're male or female, they are inherited the same way. But right now, specifically, we're looking at recessive disorders, which means you have to get a faulty allele from each parent in order for it to show up as an affected person. So if we look at something like sickle cell anemia, a person can be a carrier. These are red blood cells, as you see here. This is a normal red blood cell. This should be the shape of a red blood cell. But in sickle cell trait, what happens is the hemoglobin, the gene that causes this, causes the hemoglobin to be faulty, and that causes the red blood cell to lose its shape and take on this sickle shape, hence the name sickle cell anemia, or sickle cell trait it's called, or sickle cell disease, depending. But the problem is that if you have only sickle cells, you have trouble carrying oxygen to all your organs and organ systems, and it has multiple effects. And so it can be very devastating. But if you're a carrier, that is, you have some normal cells and some not normal cells, you appear to be normal and you can carry your blood to your tissues and be okay. So there was a study done looking at the correlation between sickle cell allele and malaria in Africa, and you can see there's a big overlap between the sickle cell allele and the presence of malaria. And as it turns out, having that sickle cell trait, that is being a carrier, is protection against malaria, which is carried by a mosquito. And part of the life cycle is that it has to spend time developing in a red blood cell. Well, if you have some sickle cells, it actually protects against malaria, which is worse than having sickle cell disease. And so as far as the theory of natural selection, those that have the best genes survive and pass them on. And so that is what is thought to be the reason that in these particular areas you see a much higher incidence of sickle cell trait in people of African descent. But in our country, it really doesn't matter whether you have the sickle cell trait or not. It's no advantage because we don't have those mosquitoes that carry malaria. So we don't have a problem with that. So we don't have any selection for or against it in our country. Hence, we don't have as many cases, but we still see it higher in certain populations. This is showing how you would do a Punnett square. This little girl has albinism, and that is an inability to produce melanin. And so when you set these up as a typical Punnett square, 2x2 box, and you can see that in this case we have two carriers. So big A is going to be normal, and little a is going to be the faulty allele. And since this is recessive, little a is faulty. And so if each parent, if there's two carriers in this, as I mentioned, is how we usually see this happening, then bring these down and across, and you can see in the homozygous dominant condition, the person is normal. But in the heterozygous condition, that person is a carrier, but only in the homozygous recessive condition do we have an affected child. So what's the chance of two carriers having an affected child? It would be 25%. So when you look on page 85 of your outline, look at some of the characteristics that we see in these people that have autosomal recessive disorders. It says most affected children have normal parents. Well, they appear to be normal, but they're actually carriers. And then it says two affected parents will always have affected children. So in other words, let me just set this up, these problems. So if this is looking at, these are looking at autosomal recessive disorders. Okay, so you put down a big A and little a, but which one, since it's recessive, little a is going to be faulty and big A is going to be normal. Okay, and so when you do, you set up your, you can have homozygous dominant, heterozygous, or homozygous recessive. In this case, if they have a dominant allele, they are going to be normal. But this person's normal, but since they have a faulty allele, we call this person a carrier, and this person would be affected. So I had a girl in my class once who was already had grown children, and she found out after, well, they weren't grown, but she had, when she found out she had tested positive for sickle cell, so we knew what her genotype was. She was affected, and her husband was tested. He was perfectly normal. He wasn't a carrier. And so she asked me, do I have to worry about my kids having sickle cell anemia? So what's the answer here? Well, here's the possibilities for this person. As we see, she's got every situation has a dominant allele. So her kids are all going to be fine, right? But instead they're all going to be carriers. So they would have to be careful if they had kids with another carrier, because again, if you cross two carriers, what's the chance of an affected child? That would be 25% chance. So again, getting back to the characteristics that you see on page 85, it says most affected children will have normal parents. Again, they're both carriers. Two affected parents will always have affected children. Well, if I cross two affected, that's all they have to pass on. And close relatives who marry are more likely to have affected children. That's why a lot of these recessive disorders show up, because remember, for a recessive disorder to show up, each parent has to have a faulty allele. So you don't see that unless, again, most of the time you're going to see that in two carriers. And we also see many of these recessive disorders showing up in royal families and things like that when there was a lot of close relatives marrying. In this case, both males and females are affected in equal frequency because it's autosomal. Autosomal means that they are inherited the same in men versus women. It's only on the sex chromosomes, pair number 23, that would be different. Okay, so that's looking at autosomal recessive. But what if we have autosomal dominant? That's even worse, because going over to page 86, in an autosomal dominant situation, you only need one faulty allele to have it. So in an autosomal dominant situation, you're still using, you're going to change this, just change all of this. Okay, so let's change this to autosomal dominant. Now, if it's a dominant disorder, then now big A becomes faulty and little a is normal. And now if you see, well, the only one that's going to be normal is if they have two normal alleles. So this person would be normal. This person would be affected. And this person would be affected. So can you have a carrier here? No. You either have it or you don't. As long as you have one, oops, did I do this right? Did this all wrong. That's faulty. Okay, here we go. This person's affected. There we go. This person's affected. And the only case you have who's normal is when you have the homozygous recessive condition. Sorry about that. Okay, so that changes everything, because now you just need the one faulty allele and you have it. So in this situation, what we usually see is, one parent will have it even if they're heterozygous, and let's say the other parent is perfectly normal. What's the chance of their kids being affected? And you can see here that, again, affected, as long as there is one big A, they're going to be affected. So at the very best, it's a 50-50. And so these are really bad kinds of disorders to have in your family. So dominantly inherited disorders are caused by the dominant alleles, and there are a lot of them here. One you may have heard of, hypercholesterolemia. This is a problem of having really high blood cholesterol no matter what you do. So you diet, you exercise, do everything right, you still have high blood cholesterol. This is a genetic disorder, and what happens here is you just don't produce the receptors to clean out your blood as you should and get the cholesterol out of the blood and into the cell where it should be. So here's a clip on that one.
Familial hypercholesterolemia, or FH, is a genetic disorder characterized by greatly increased LDL levels and a family history of premature atherosclerosis. Normally, LDL receptors on liver cells help clear LDL particles from the bloodstream. FH may be caused by any of over 1,000 specific mutations that affect 3 known genes. More than 80% of these mutations increase LDL levels by causing the LDL receptors on hepatocytes to either be functionally altered or underexpressed, decreasing the body's ability to clear LDL from the bloodstream. The result of either scenario is an increased plasma concentration of LDL particles. In addition, impaired LDL uptake also decreases the cholesterol pool in the liver, which stimulates hepatic cholesterol synthesis and the production of VLDL, a precursor lipoprotein to LDL. These defects found in FH can result in an increased concentration of LDL particles starting early in life. sustained exposure to high LDL levels, and increased deposition of cholesterol into the artery walls over time elevate the risk of atherosclerosis and premature cardiovascular disease associated with FH, making early detection of this genetic disorder of critical importance.
So usually if you have something like that in your family, you are going to have to go on some kind of medication to control that, so your blood levels of cholesterol. Achondroplasia is a form of dwarfism, and that is another example of a dominant disorder. So again, in this case, I just did this on the board, but if you're looking at, remember, keeping in mind that the dominant allele is the faulty allele here. And so here you have a perfectly normal person with a dwarf parent. And here's the thing about this, these parents, in this case, they know they have it. So what's the chance if they have kids with a normal height person of having a dwarf? And again, that's 50-50 at best, assuming the other parent is perfectly normal. So this is a case, and not all dwarfism, by the way, is autosomal dominant, but this is a case that you would see normal, kids from dwarf parents possibly showing up. When we look at the characteristics for autosomal dominant inheritance on page 86, it says an affected child usually has an affected parent. Well, wouldn't you think an affected child always had an affected parent? Think about this. There is a case of progeria, it's called. Progeria, which is premature aging, and this is also an autosomal dominant disorder. And in this case, these kids age and die before they would ever reach puberty, so they will never have kids. So where did they get their faulty allele? So here's the thing. Sometimes these alleles, they have to start somewhere. Sometimes they arise just out of the blue through mutation. The thing is, once you have it, then the question is, what's the chance of passing it on? But it doesn't mean you can't get it even if it's not in your family, because some people are just born with these mutations in the first place. So my point is, it's not always your parents' fault. It may be that it just cropped up, which it does with all mutations. Mutations are spontaneous changes in DNA, and they just happen spontaneously, so you just don't know. So when it says an affected child usually has an affected parent, unless it's something like progeria. Homozygous dominants are rarely observed. Why? Because that would say both parents had a faulty allele for it to be homozygous dominant, which means both parents have the disorder. And if two parents know they have these situations that are genetic, they usually don't have kids with the same. Unaffected persons do not transmit the characteristic. Again, if they've got the normal situation here, they're not going to pass it on. And as with the other, since it's autosomal, both males and females are affected with normal, with equal frequency. Now, another one is Huntington's disease, and this one's really bad. It's a degenerative disease of the nervous system. And so, as you can see in this chart, that even though you've inherited the gene, the actual symptoms don't really start kicking in until you're in your 30s and 40s. That's when you start showing all the symptoms exponentially, as you see here. And oftentimes, people have already had their kids by then. And so the question is, what do? There is a test for this. And so if you already know you have Huntington's, but your kids don't know if they have it, and they want to be tested and they find out they have it, well, what does that tell you? So you have it as well. So there's certain situations where genetic testing gets kind of um, there's some ethical concerns, because if the kids show up, get tested, and they have it, then that tells the parents they have it. Maybe they didn't want to know because they already had their kids. Who knows? But here there is some hope with this one. There's some new options in gene therapy, hopefully, that will help this, because it's a lot like Alzheimer's, only it lasts a lot longer. And other than that, though, it's a degenerative disease of the nervous system, which is really, really just a sad, sad situation to be in. So here's a clip on that.
Huntington's disease is a genetic brain disorder that has a widespread effect on how our bodies function. Mutations in the Huntington, or HTT gene, causes a progressive breakdown of nerve cells, or neurons, located in the brain. This eventually affects an individual's ability to move, their mood, and even how they think. The exact function of the HTT gene in healthy adults is not yet fully clear. But as an adult, the inherited mutation in the HTT gene leads to the production of toxic protein that causes the death of neurons in the brain. This disease is devastating to the affected person and is extremely difficult for their family too. As independence is slowly lost, it is hard for caretakers to manage both their emotional and medical needs, not to mention the stress related to the potential risk of the disease being passed along to their children. This is a 50-50 chance. However, gene therapy might offer hope for this disease, which currently has no cure or effective treatment options. Gene therapy typically changes how cells produce important proteins by replacing a gene that is missing or abnormal. In the case of Huntington's, gene therapy would actually help lower the cell's production of the toxic HTT protein, ideally slowing the breakdown of neurons in the brain. The two main approaches currently in clinical trials use either microRNAs or antisense oligonucleotides to accomplish this. To talk more about microRNA, here's Dr. Beverly Davidson.
MicroRNAs are short RNA molecules that prevent or reduce the production of a particular protein. MicroRNAs work as a check and balance system to control protein levels by binding to and destroying the messenger RNA that would have produced that protein. As an example, we can reduce the level of toxic proteins that are causing Huntington's disease by using gene therapy to insert microRNAs to bind to and inhibit production of the mutant Huntington protein.
The microRNA needs to be delivered into a cell by a vector, which is often derived from viruses. Don't worry, all viral genes are removed, and the vector is modified to only deliver therapeutic genetic material. The modified cells with the microRNA provide instructions to produce much less HTT protein. The other approach, antisense oligonucleotide, referred to as ASOs, is an antisense therapy that is designed to block or alter how cells express specific genes. Antisense therapy uses synthetic DNA or RNA mimics that bind to the faulty HTT gene to block its ability to produce the harmful protein that causes the death of neurons. A key difference between these two techniques is that antisense oligonucleotides typically require multiple doses, while microRNAs aim to be administered only one time. Researchers continue to work optimistically in clinical trials to safely and effectively slow down the progression of Huntington's disease. For more information and resources about gene therapy for Huntington's disease, go to asgct.org.
Okay, so we actually are going to talk more about gene therapy and those things when we get to the chapter on DNA. So we'll come back to that later. But the last thing I want to do is just talk a little bit about some homework and some labs that you should now be able to do that are uploaded on Canvas. First of all, the genetic practice problems, make sure you practice those, page 87 and 88, page 96 in your course outline. Now, we haven't gone over page 96 yet, or those problems. That's actually in our next chapter that we'll be covering, chapter 15. But we'll get to those. Make sure you do all of these and practice them before you take your next test. Remember, the solutions are in your course outline, and the actual Punnett squares are on Canvas under the Canvas module handouts. So practice those. Lab 10, meiosis. This is a computer-assisted lab, and the goal is for you to learn the stages of meiosis, and we've been over those. So what you should do is go to Canvas, modules, lab 10, click on the tutorial, complete the biology project tutorial and the problems, and make sure you record your answers on your printed quiz in your lab manual so that you can use that as a reference for taking your online quiz. And then take lab 10 online quiz, and that's how you will get your 10 points for the lab 10. Also, just a note here, there's a mistake on the quiz question number 6, make sure you make this note. It should read, that is the quiz question in the actual biology project tutorial. I can't edit that, so that's why I'm just telling you now to make a note of this. For question number 6, it should read the stage of meiosis where homologous pairs of chromosomes separate. That's what you should have there. So, and I did go over this. I'm not sure if I went over it with both classes, but okay, so that's meiosis. That's lab 10. Now, lab 11, let me talk a little bit about this. So, in this case, we would normally be counting some corn seedlings for the first part, and then we would be counting some seeds on genetic corn ears in a second part. But instead, I'm actually giving you the data here and also when you go online. So this is all online now. So when you go online, I'm actually giving you the data, and this is under the O column for observed. Now make sure you read this lab because it walks you through these calculations so that you know how to do them. I'm not going to do all the calculations because they're explained in your lab manual, but I am just, kind of explaining what these columns are. So here's your O. This is if you are actually counting what you would observe. So then what do you expect? If you add these two up, that would be N, total number of plants. And let's just say it's 100. So what you expect is this is your 3-to-1 ratio. You expect 75% to be green and 25% to be albino. So you take 3 times N. Remember, this is N, not this, but total plants. 3 times N divided by 4 goes here. N divided by 4 goes here. These two columns should add up. If they don't, you did something wrong. That's your check. Then you subtract the 2, O minus E, put that here. If you get a negative number, it doesn't matter because then you're going to square it. And when you square a negative number, of course, it becomes positive. Then you take this column divided by the E column, so this column divided by this column, and that gives you your last column. Add that up, and that gives you your chi-squared total. Now, your degrees of freedom are defined as the number of data sets minus 1. You have 2 data sets, green and albino, minus 1. That would be 1 degree of freedom. Then you have to go in your table of critical values that are in your lab manual and look up under 1 degree of freedom, accepting 5% error, and look up that number. I'm just going to guess off the top of my head it's 3.82. Might be, might not, I'm not sure, double checking. But anyway, as long as this number is less than this number, the data does fit. So down here, since my calculated value, whatever you calculate here goes here, is either more or less than your critical value of whatever you look up in the table here at 5% error. And by the way, we have to decide how much error we're accepting when we make these statistical probability calls. And so we're in other words, we're saying we're going to be 95% certain that this ratio is a 3-to-1 ratio. Remember, this is the monohybrid cross. We're expecting a 3-to-1 ratio. This is dihybrid where we expect a 9-3-3-1 ratio. So here's 3-to-1, here's 9-3-3-1. So again, back here, since your calculated value, whatever you have here, is more or less than the critical value, whatever you have here, as long as it's less, the data does fit the predictive ratio. Again, this is all explained in your lab manual. Then over here, we give you more data. Again, these are the kernels that we observed. So you add those up. There's your total kernels. That's N. We're expecting a 9-3-3-1. So 9 times N divided by 16 goes here. 3 times N divided by 16 here. These two numbers should be the same. And the last one is N, this number divided by 16. Add them up. These two columns should be equal. That's your check. Subtract the 2, that goes here. Square it, that goes here. Divide by expected, that goes here. Add this last column up, that goes here. Degrees of freedom are going to be 4 data sets, minus 1 would be 3. Look up your 3 degrees of freedom in your critical value table at 5% error, and there should be a number here. And I'm going to just guess it's 7.82, but I don't know. I'm just doing this off the top of my head. So you guys Look that up. It is 7 something. I don't remember exactly. All right, since my calculated value of whatever you get here goes here, it's either more or less than your critical value of whatever it is you get here goes here. If this number is less than, since your calculated value, whatever that is, should be less than your critical value, then it does fit. And then here you want to do a Punnett square. I helped you set this one up. I didn't finish it for you, the dihybrid cross. The last thing in this lab is looking at blood types. Since you're not going to be able to actually do the typing, you can look at those and figure out who's who using this chart. And then you have to explain who's the daddy based on who can't be the dad based on these blood types. And that goes here. And then you take your online quiz for that, and that will give you 10 points for the genetics lab 11. Okay, last thing, directions for completing lab 12. I'm going to give you these now, although I haven't really talked about the karyotyping lab yet, but in case anyone wants to get an early start, this is another simulated lab. Go to your Canvas modules. There's directions there. It tells you to run the karyotype activity, answer the questions on your lab report, and then take the quiz. So I'll explain more about this when we get to chapter 15. All right, folks, I know that was a long time. Remember, you can always pause during these videos to take a break. And now it's break time before we come back to chapter 15 next time. You guys have a great day and be well.